I'm Michelle Boxall and welcome to Behind the Eight Ball. When the odds are stacked against you, how do you get out in front? Well, in IBTV's flagship series, we will hear about the failures along the road to success for many of the industry's CEOs, leaders and founders. We hope we can all learn from these stories and that you'll enjoy them and that they'll provide inspiration for new and future successes for the industry. So with great pleasure, I'd like to introduce Dr. Bruce Levine. We're super excited to have an ISCT and cell and gene therapy industry giant. You're very welcome.
Well, thanks, Michelle. It's a pleasure to be here with you.
So Bruce is one of the founding directors of the Clinical Cell and Vaccine Production Facility at the University of Pennsylvania. He's the co-inventor of the first FDA-approved gene therapy, Kymriah, which was licensed to Novartis, and co-inventor on 32 issued US patents. Looking back, what do you think set you along this path and onto so many accomplishments?
Yeah, I think it's being in the right place at the right time and being part of a great team. I did my graduate work here in Baltimore at Johns Hopkins University and went to do a postdoctoral fellowship with Carl June at the Naval Medical Research Institute. And between those two stops, learned a lot about T-cell biology and how to culture T-cells. And in a postdoctoral fellowship where I have the opportunity to create a new way to grow T-cells that turned into how we grow T-cells today in many adoptive immunotherapy clinical trials, not just at our center at Penn, but it's being used around the world.
Wow. And so looking back in your kind of formative years when you're a young adult, what was your approach to failure? Were you worried about it? Was it something that ever crossed your mind?
Yeah, I think it's the rare person who's not afraid to fail, because if you're not afraid, maybe you're engaging in things that are a little dangerous or maybe foolhardy. But being a scientist, you have to be prepared to fail. That's why they call it research, right? It's not search. You're searching for something, it doesn't work, and then you search again or you tweak something. So we had a lot of that along the way. And just in the first clinical trial that we were gearing up to do, this was in HIV positive subjects. And to do that, we had to figure out a way to grow T cells from HIV positive people. Now, at the time, and this is in the 90s, you couldn't do that because if you did activate T cells, then the activation of the T cell would release a whole lot of HIV and destroy the cell culture. So one way that we developed was to say we're going to stimulate T cells with these beads. I'll describe in a moment. We'll put antiretroviral drugs in the culture media, and that will prevent HIV from overwhelming the culture. And then Carl said to me, well, do it in a condition without the drugs so that you can show that the drugs are working to control the virus. Now, how do we stimulate cells? This was mimicking dendritic cells with a tiny magnetic bead to which we conjugated two antibodies, anti-CD3 and anti-CD28. So I do the experiment, both conditions, with drugs, without drugs, and there was not HIV in either condition. So I take that result to Carl. He said, you're doing it wrong. I said, well, I know I calculated right and I added the drug, and in one I didn't add the drug. And I only found out maybe over a year, a couple years later, when I was home visiting my parents for a long weekend, he repeated the experiment behind my back. And he got the same result. And it turns out, we didn't know it at the time, but when you use these beads, co-stimulating T cells, anti-CD3 and anti-CD28, it downregulates the co-receptor that HIV needs to get inside the cell. So we were inducing an anti-HIV state in these T cells, and that's why we didn't see the virus come out. So That then became a couple of science papers and more follow-up papers, and also enabled us to conduct that clinical trial in HIV-positive people, where we showed that we could increase CD4 counts and immune function.
Oh, that's really interesting. So moving on from your approach to failure, can you talk us through your behind the eight ball story?
Yeah, so that was a lesson in itself. I thought that I had failed, right? But then it goes on to that enables us to conduct the clinical trial. Now, we were at the Naval Medical Research Institute, and we were able to do trials in HIV, but you can imagine with an approach from failure, it does or it doesn't
Yeah.
suppress HIV. But now there's other opportunities that come out of that. So from failure comes opportunity. And the opportunity was to do this not only in HIV, but in cancer. And Carl June at the time had been recruited all around the country, maybe internationally, and then opportunity came up at the University of Pennsylvania, where I had gone as an undergrad, and I then had the opportunity to set up the lab that we had at the Navy at Penn, and then that now has grown into many, many clinical trials over the years, and a large team of people that enable clinical trials in cancer and HIV, and now we're talking about other applications. So if at first you think you made a mistake by not adding the drug when you really did, it turns out to be true that you did add the drug, but a surprising finding. So I'd say that sometimes things don't work. Sometimes things do work, and sometimes there's a surprise.
Yeah.
And the really interesting stuff comes from the surprises, because that leads to many other potential areas of work.
Yeah. I've heard it so consistently speaking to the leaders in the series that often through a failure, seemingly a failure, you learn something along the way. And actually, without that failure, you wouldn't have gone on to the successes.
Yeah.
There's something significant being learned.
Yeah.
Yeah.
Yeah. So sometimes things go right, sometimes things go wrong, sometimes things go strange,
Yeah.
and you learn from the wrong things. But what's really interesting is the unexpected result.
Well, when I was talking to Luigi Nardini the other day, he was talking about curiosity, and I think I could see that in this situation, in that some people may have gone into that situation, seen a failure, and not maybe thought they were going along a wrong track, where actually you're trying to find out why, and that's then led you to uncovering
Yeah.
the surprise.
Well, there's an experimental checklist, right? So if something doesn't work the way you expected, you go down the checklist and say, did I do this? Did I do that? Did I do the other thing? And you try and figure it out. And if it's none of those, it must be something else. And that's where the discovery comes into play.
So moving on from your behind the eight ball story and on to your success with Kymriah, could you tell us a little bit about how you got there?
Yeah, so we had actually been, based on the success in being able to grow T cells, begun a collaboration with a company called Cell Genesis, which was the first company ever, the first group ever to conduct CAR T cell clinical trials. And this was in HIV, actually. So we continued that after we first moved to Penn. That was in 1999 to 2002. And then in 2004, we got funding from Philanthropy, the Alliance for Cancer Gene Therapy, to begin our preclinical work on CAR T cells in cancer. We treated our first patients in 2010. We had 2 complete remissions and 1 partial remission in the first 3 patients that had failed everything under the sun. Ran out of money, maybe that's another 8-ball moment. Then what do you do? Because it's supposed to be a 20-patient clinical trial. And Carl June and David Porter decided, well, let's just publish what we have, and maybe that will turn out well and lead to grant funding or other funding. Those papers came out in August 2011, and then there were entities that came to Penn wanting to license the technology. One of those was with Novartis. We signed the alliance with them in 2012, and in the interim had treated the first patient with CAR T cells in the world, Emily Whitehead, and today is actually the 12th anniversary of her cancer-free day. So that led to us transferring the technology to Novartis, working with them to establish the methods for manufacturing and analytics of CAR T cells and conducting joint clinical trials. And in 2017, that led to the first FDA-approved drug, CAR T cells for pediatric acute lymphoid leukemia, and that became Kymriah.
Amazing. Um, and with that success, often you look at leaders and think, well, there's something about you that was always going to be successful. But in getting there, you obviously need to be a leader. How do you lead and inspire a team? Or what type of leader are you when working with others?
Yeah, I think in our case, it's focusing on the mission and on the application. The science is there, and then the challenge is how to translate to clinical trials and bringing that back to the people doing the work, right? Why are you doing the work? Well, would you like to bring the cells that you made to the infusion site to meet the patient? And what we found over the years is having the patients come and tour the lab and meet the people in the lab gives them great inspiration, but that then gives inspiration back to the patients. So it's a real 360 in that way. We always emphasize, why are you doing the work? It's to benefit patients who have no option. And meeting patients and families really has been a blessing in the work that we've done.
That's inspiring. Um, in terms of, obviously, you've been in this industry a long time, a huge leader. What would you, if you could pick one piece of advice for industry, what would that be for others coming up the ladder?
Yeah, well, of course, there's the persistence and working hard, but I think keying in where we are at this meeting and in a couple of weeks at the ICT meeting, it's to network. Don't be afraid to go up to people, to meet them, introduce yourself, present your work, whether it's a post or an abstract, take invited talks, and then those lead to other opportunities, the awareness of your work, but also other opportunities. And then that leads into the funding mechanisms, whether it's grants or philanthropy or industry alliances.
I think at ASGCT, that's been a big focus. I keep hearing about networking, and I think this industry in particular is a warm one. Everyone seems open to discussions, which is great. Um, and then final question: how have your ambitions evolved over the years? Are there more things? Are there things that you're looking forward to doing? You've got other goals.
Yeah, well, now focused on new technologies. So we have the first generation of products approved, but we need to improve them so that we're able to induce remissions in more patients than we are currently. We're moving from the so-called liquid tumors, leukemia, lymphoma, myeloma, into solid cancers, and again, into other applications. How do we enhance potency? And then in my role at ICT and in other roles, I'm looking at how can we bring CAR T cells to other regions, whether it's rural areas or areas across the world. And in the past year, I've been to India and Brazil and other countries looking at establishing CAR T cell manufacturing and other advanced therapy, cell and gene therapy. I'm actually now working with a cohort of Wharton MBA, executive MBA students, and their project is to assess where is it possible to bring CAR T cells in Africa. And the approach we took is, where do they have transplant programs? So we work from there, from the stem cell transplant programs. And then finally, it's on mentoring. We have really, really bright students and fellows, at the University of Pennsylvania, and I've had a chance to get to know some of them. They're working on incredible stuff, and it's telling them, okay, you're at the early stage of your career. What is your project about? What would you like to do? And helping them along their way to develop in their careers.
Sounds super exciting. I have to keep checking in and find out how that goes up. How that works. So thanks, Bruce, for your time. Really appreciate it, and had lots of learnings and insights. And I hope we'll see you again.
Yeah, thank you.